Okay, so let's move on to section 4 of chapter 6. I'm going to break this up into two different lectures because it's probably one of the longer sections in this chapter. So just as a reminder, bones are organs, and we talked about osseous tissue in the connective tissue section of the tissues chapter. But even though bone is made up of osseous tissue, it also has other tissue types as well, including nervous tissue, cartilage, fibrous connective tissue, muscle tissue, and even epithelial tissue that's making up the blood vessels that are supplying it. When we talk about the structure of bone, there's three levels in which we can talk about it. Gross structure, those would be structures that we can see with our naked eye. We also have microscopic structures, which we would need a microscope to identify, and then the chemical composition or structure of bone. So we're going to start with gross anatomy. And the biggest thing here is basically what we can see with the naked eye are two types of bone. We can see compact bone or spongy bone. And compact bone is going to be your external layer of every single bone, and it's going to appear smooth and solid. So if you look at any bone in the lab or know what a bone looks like, everything that you can see externally on that bone is compact bone. In order to see the spongy bone, we'd have to actually cut the bone open. And if we cut the bone open, we might see spaces or regions like this, which are not compact. And this would be your spongy bone. And spongy bone is made up of these lines, if you can see it at all. All of these lines are bone, but we specifically call it trabeculae. And within the trabeculae, we have these spaces. So all these little holes right here between the trabeculae are going to house some marrow. This right here says red or yellow marrow, and that's basically going to be dependent on what age you are. So spongy bones on the inside, it's made up, like I said, of trabeculae. We have compact bone everywhere on the external surface from what we can see. So when we're talking about compact bone and spongy bone, we have different compositions depending on what shape of bone we're talking about. So our short, irregular, and flat bones have a specific composition of spongy bone and compact bone. So basically all the bones that are not long bones. And then long bones have a different composition. So we're going to look first at the short, irregular, and flat bones. And basically what it is we have a layer of spongy bone, and on either surface of the spongy bone, we have compact bone. We also have to bring back to play our membranes. So we have our periosteum, which is going to be on the outside or external surface of the compact bone, and our endosteum, which is going to cover virtually every space within the side of the bone. We do have some bone marrow scattered throughout the spongy bone. Those would be in those spaces between our trabeculae, but we don't have a defined medullary cavity or a marrow cavity. Only would we have a medullary cavity or defined marrow cavity if we're talking about a long bone. And if we're talking about where these bones connect at a joint that is movable, it's going to be covered with what we call hyaline cartilage. So here's a good image of what a typical structure if we're talking about spongy bone and compact bone of the short and irregular and flat bones, where we have this layer right here of compact bone, then we have this layer of spongy bone, and then on the other side of the spongy bone, we have more compact bone. So here and here, we would have the periosteum or the outer membrane. And then lining all of these trabeculae and all of these cavities on the inside, we would have endosteum. So down here, they've taken this area and zoomed in. So we can see all these bone spicules right here. All of this is trabeculae. And here's our holes between the trabeculae. And this is where our marrow would be right there. So that's the structure of everything except for a long bone. A long bone's a little bit different because we have parts to a long bone. So we have what we call the shaft or the diaphyses. That's going to be the long axis of the bone. And then we have the epiphyses, which make up the ends of the long bones. So the diaphyses is going to have a central space, which we call the medullary cavity, and that's going to be filled with yellow marrow only if you're an adult, and we'll talk about that in a little bit. Our ends of our long bones or our epiphyses, they're a little bit different. because they have an external layer of compact bone, but internally they have spongy bone. And most of these are connecting or are a component of a joint surface, so we're also going to have some cartilage there. So here's a better picture where we can see our actual long bone structure. And again, this is our shaft, or what we call the diaphyses, and then our two ends, which we call the epiphyses. We can label these ends using our directional terms. So we see proximal here. So we know proximal from chapter 1 means closer to the point of attachment. So this would be the one closer to the point of attachment, while our distal end would be the one farthest away from the point of attachment. So we can see in our diaphyses, it's predominantly this giant space right through here, and this giant space is what we call the medullary cavity. And the medullary cavity is going to house bone marrow, and right here they're showing you a G yellow, so that is going to be in an adult. The ends or epiphyses are a little bit different because we have an outer layer of compact bone, which you can see here because we don't see any trabeculae. However, the internal surface, or sorry, the internal space of the epiphyses is all spongy bone. And there's no defined medullary cavity at all, even though we can house some bone marrow in those spaces between the trabeculae. Another thing I want to point out is this line right here. It looks like it's compact bone between the shaft, the diaphyses, and the epiphyses. This is what we call the epiphyseal line, and this is what we would get compact bone in that space if we're done growing. If we're still in adolescence and we are growing in length still, what that is called is not the epiphyseal line, we call it the epiphyseal plate. And the epiphyseal plate is made up of cartilage. So remember, the only way to produce new bone is to actually grow it from cartilage. So the epiphyseal plate in an adolescence is cartilage. Once your epiphyseal plate is no longer cartilage and it's converted to bone, you can no longer grow in height, at which point we call it the epiphyseal line. So here's just if we're going into one of the ends or the epiphyses of a long bone, we can see we have the external surface of compact bone right here. And then predominantly the whole inside of the epiphyses is all made up of spongy bone with these trabeculae, or these bone spicules, and the spaces right here that are going to house bone marrow. So let's talk a little bit about the membranes. I think I mentioned previously the periosteum, or the outermost membrane, but it is a double layered membrane. So we have two layers to the periosteum, or the membrane covering the surface of compact bone. The outermost layer of the periosteum is what we call the fibrous layer. It's made up of dense, irregular connective tissue, but what's most important is that it has these protrusions off of the fibrous layer. And these protrusions off the fibrous layer are what we call perforating fibers. And these fibers are going to help adhere the periosteum to the compact bone itself. The inner layer of the periosteum is called the osteogenic layer because it contains what we call osteogenic stem cells, so basically bone cells that can give rise to other types of bone cells. We're also going to see these little holes that we call nutrient foramens. And what these holes or nutrient foramens are is they're pathways for nerve fibers and blood vessels as well. And the periosteum can also provide anchoring points for tendons and ligaments. In contrast, we have the endosteum. It doesn't have to be as tough as the periosteum because it's not on the external surface. This is just the membrane, remember again, that's lining basically every internal surface of the bone. So what is it going to cover? Things like the trabeculae, all of the canals. There's going to be two canals that we're going to talk about. Everything, every space, cavity, hole, canal, anything on the inside of bone is going to be lined with endosteum. And like the periosteum, it also contains the osteogenic cells that can give rise to other types of bone cells. So here's a really good picture of the diaphyses of a long bone, so the shaft, and we can see the medullary cavity right here. In this case, it's housing yellow marrow, so that means we're looking at an adult. We can see these holes that are going horizontally through the axis of the bone. These are nutrient foramens, and they're going to allow passage of our nutrient artery. Also not shown is our nutrient vein and some nerves going through this foramen as well. They're just showing you the nutrient artery though. We see these perforating fibers, so all these white things right here. These are your perforating fibers, which are extensions of your periosteum. And remember, those are going to help adhere the periosteum to the bone itself. Now we can talk a little bit about hematopoietic tissue in the bones. So primarily when we talk about hematopoietic tissue, we're talking about red marrow. The biggest difference here is that if you're still growing and you're an adolescent, the bulk of your marrow is going to be red bone marrow. When you're an adult and you have stopped growing, the bulk of your marrow is going to be yellow marrow. So, housed in our medullary cavity of our diaphyses or shaft of our long bone, that what I pointed out earlier, that yellow marrow, because it's yellow marrow, it means that you're an adult. And basically, the yellow marrow is just like a storage, a fat storage, or energy storage for us. However, in the event, say we become anemic and we need to synthesize more red blood cells, we can convert that yellow marrow back to red marrow. But if you're a healthy adult, you have like 90% or more yellow marrow and very, 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 very little red marrow. And where would we find the red marrow? We could find small amounts in some flat bones, some irregular bones, but in the heads of the femur or humerus. But again, the bulk of the marrow in adults is yellow, and that of an adolescent or a child still growing would be red marrow. So now that moves us on to bone markings. So these are things that you can see. Again, we're still under gross anatomy with the naked eye. We lump them into three types of bone markings. We have projections, we have surfaces, and then we put these two into one category, depressions and openings into our third category. Why do we put depressions and openings and lump them together? Is because they both act as passageways for blood vessels and nerves. When we talk about a projection, what is a projection? It's an outward bulge of bone, and this is going to be sites where our muscle and ligament attachment can occur. And when we talk about surfaces, these are going to be areas of the bone that actually form the joints. So table 6.2, you definitely need to study. You should be able to describe the three categories that we talked about, and that's how this is broken up. So this table on this slide is showing you specifically projections, and that these are sites of muscle and ligament attachment, just as a reminder. So we have several types of projections. We have tuberosities, crests, trochanters, lines, tubercules, epicondyles, spines, and processes. You should recognize that all of these are forms of projections, which means that all of these are sites of muscle and ligament attachment. Additionally, you need to know what their description is. And again, this is just a general description for each one of these bone markings. This is going to help you in lab because in lab, you have to identify the specific name of each bone marking. So for example, not only do you need to know that this is a tubercle, but specifically this is your adductor tubercle. So this gives you an idea right here. If you know that a tubercle is a small rounded projection or process, now you know what to look for over here. So this will help you in lab, but again, you do need to know what each of the projections can be described as. And then in lab, you have to identify the specific name of each projection. So here we can see our surfaces. Again, these are areas that help form joints. We have three types of surfaces. We have heads, facets, and condyles. So basically understand their basic description and that all of these are in areas at which joints are formed. This is a head. Here's a facet. We can see a condyle right here. And same thing for depressions and openings. Again, understand that all of these are depressions or openings, which means that they are passageways for blood vessels and nerves. And then understand their general description. So, for example, you will definitely need to know that a foramen is some sort of round or oval opening through a bone. When you hit the skull, I think there's at least 8 different foramens that you need to know specific names of. So foramen spinosum, foramen rotundum, foramen magnum. If you know what a foramen is and it's an opening, then it'll help you identify these specific structures or these specific bone markings.